Morning. For those of you who don't know me, I'm Richard Mann. I'm a professor of biochemistry and neuroscience at the Zuckerman Institute. And it's my pleasure today to introduce to you Dr. Vanessa Ruta, who's currently the Gabrielle Ream and Herbert Caden Associate Professor at Rockefeller University. Vanessa is a true New Yorker, having spent almost her entire life on the island of Manhattan. Yet her path to becoming a Rockefeller professor was not completely linear. She spent 4 years as a professional ballet dancer after graduating from Stuyvesant High School. After that detour, Vanessa obtained her undergraduate degree in chemistry at Hunter College, and then from there did her PhD at Rockefeller University, where she carried out really elegant structural and biophysical studies on voltage-gated potassium channels with Rod McKinnon. From Rockefeller, Vanessa moved uptown, where she was a postdoctoral fellow with our own Richard Axel. And while in the Axel lab, where I first met her, Vanessa began to work on Drosophila and carried out beautiful work characterizing a neuronal circuit that allows a sex pheromone to elicit sexually dimorphic behaviors. Since moving back to Rockefeller in 2011, Vanessa continues to distinguish herself by using an unusual combination of biochemistry, structural biology, and Drosophila genetics to understand how neural circuits mediate the fly sense of smell, are wired, and flexibly interpret and respond to complex environments. Vanessa has already been the recipient of several prestigious honors, including the Harold Weintraub Graduate Student Award, an NIH New Innovator Award, and awards from the Pew, Sloan, and McKnight Foundations. And the title of Vanessa's talk today is Circuit Mechanisms for Behavioral Evolution. Please join me in welcoming Vanessa.
Thank you very much for that introduction, and I just want to say what a great honor it is to be here, to be participating in the Inaugural Symposium, particularly because I'm so indebted for my time as a postdoc in the Axel lab at Columbia, and really the rich environment actually on the Hammer 10th floor, which was created by Richard and Tom. And I just want to echo actually really what Catherine said. I think that was a completely transformative experience and really made me into a neuroscientist. So I'm very happy to see that actually the scientific community of Columbia is now actually housed in a building where the splendor and beauty really matches the science. So animals exhibit an extraordinary diversity of behaviors, both within and across different species. And while I think we have an increasing appreciation and insight into really how circuits can adapt rapidly through learning and experience to allow for behavioral variation at the level of individuals, we know far less about how circuits are adapted over these longer time frames, over evolutionary time scales. And so one question my lab has become interested in is really how is it that evolution shapes the nervous system to give rise to the incredible variation that exists across different species. And a few years ago, one approach we thought we could take is to begin to really directly compare the homologous sensory processing circuits in closely related Drosophila species to begin to really pinpoint sites of evolutionary adaptation. And so the work that I'm going to describe today was really spearheaded by one of my first graduate students in the lab, Laura Seeholzer. And the circuits that we decided to look at, which David already introduced, are those which underlie male courtship. So in Drosophila, male flies court females with this sort of elaborate ritual in which a male orients towards a female, taps at her abdomen with his foreleg to sample pheromonal cues. And if this discrete gustatory assessment indicates that she's an appropriate mate, as it has in this case, you can see that he will persistently pursue and follow the female, extending a single wing and vibrating it to generate a species-specific courtship song. So one of the really compelling aspects of this behavior is the fact that it is entirely genetically specified and hardwired into the male's brain. But another reason, of course, that these courtship rituals were very interesting to us is the fact that they evolved very rapidly as a mechanism to reinforce the reproductive boundaries between different species. So for example, Drosophila simulans is a sister species to Drosophila melanogaster that diverged only about 2 to 3 million years ago. These two species meet each other often in the environment, yet they remain reproductively isolated. Copulation between them is rare, and when it does occur, they produce offspring that are sterile and only of one gender. So what are the neural mechanisms that prevent this kind of futile copulation? Well, one mechanism is actually males are quite choosy in which females they're willing to court. And this is apparent from preference assays where we offer males the opportunity to court females of either species. So here, for example, you can see a movie of a simulans male that is in the process of courting its conspecific simulans female. It pursues her, follows her, becomes temporarily distracted by the presence of a melanogaster female who he goes over to investigate and assess and quickly rejects and returns to his first love. So you can see Drosophila simulans are highly selective and will only court Drosophila simulans females, whereas Drosophila melanogaster males, although a little bit more promiscuous, still also strongly favor courting Drosophila melanogaster females. And we think that this kind of discrimination at the level of the males is due to the fact actually that females of these two species actually carry different hydrocarbon pheromones on their cuticle, with melanogaster females making one compound called 7,11-heptacosadiene or 7,11-HD, whereas simulans females make a different compound called 7-tricosine, which is actually the same compound that is carried by simulans males and melanogaster males. And actually work from Joel Levine's group about a decade ago suggested that this pheromonal difference at the level of the females is actually sufficient to generate a barrier to courtship and copulation between these species. So what he showed is actually if you just take a simulans female and now perfume her with a melanogaster pheromone, you now render her highly attractive to the melanogaster male, but really repellent and repulsive to the simulans male. And so what this experiment suggests is actually that males of both species can actually detect the very same pheromone, 7,11-HD, but they endow it with the opposite interpretation in the context of courtship. In one species, it promotes courtship towards an appropriate mate, and in the other species, it suppresses courtship behavior. And so we're interested to ask where in the nervous system have changes occurred that allow for this opposing behavioral interpretation. And in particular, is it at the level of the sensory periphery in the way this information, the pheromone is detected, or is it at downstream central circuits in the way this information is processed by higher brain centers? And we were particularly interested in looking at the sensory periphery in part because of the idea that this is likely to be the most evolutionary labile portion of the nervous system because it is freed from the kind of complex developmental constraints that are found in central circuits. And so, as I mentioned, these pheromones are initially detected in an early component behavior of courtship where a male actually extends its foreleg and samples the cuticular hydrocarbons on the female. And there are multiple classes of sexually dimorphic gustatory sensory neurons on the male foreleg, including one large population which expresses an ENaC channel called PPK23, and in melanogaster males is thought to detect the melanogaster female pheromone to promote courtship behavior. So we're interested to ask, what is the role of this receptor in Drosophila simulans? And so to do this, we use CRISPR-Cas-mediated mutagenesis to make a null allele of this PPK23 receptor in Drosophila simulans, and then examine the consequence and preference assays. And what you can see is that actually the loss of this single receptor makes simulans males completely indiscriminate. They are unable to distinguish between simulans and melanogaster females. And we think that this promiscuous kind of courtship behavior, this lack of inhibition to the melanogaster female is actually due to the fact that they can no longer detect 7,11-HD. And this is because we can actually carry out preference assays where we now offer males the opportunity to court a simulans female or a simulans female that is perfumed with a melanogaster pheromone. And you can see that wild-type simulans males will actively avoid courting these perfumed females, but PPK23 mutants are basically unable to distinguish between these two females. So this suggests that actually males of both species, we think, rely on the very same receptor, PPK23, to detect 7,11-HD. And from this and other experiments suggests that actually it's not likely to be a change of the sensory periphery that underlies this opposing behavioral response in these two species, but really is due to differences in downstream central processing pathways. So to gain access to these central circuits, we took advantage of the fact that actually almost all aspects of male courtship behavior in flies are specified by the male-specific isoform of the fruitless transcription factor, which is expressed in only about 2,000 neurons that are distributed throughout the central nervous system. And actually really beautiful work from a variety of different labs has suggested that these 2,000 neurons form interconnected circuits that underlie almost all aspects of courtship behavior, everything from sensory detection all the way through to motor implementation. So to gain access to these same circuits in Drosophila simulans, we used homologous recombination to knock into the simulans genome and label the fruitless positive population. And you can see that it labels an anatomically very similar ensemble of neurons, really then providing us with an inroad to make direct comparisons in the anatomy and function of the neurons in the pheromone processing pathways. And so the first thing we wish to do is really to provide functional evidence that actually the sensory periphery is conserved. So we expressed the genetically encoded calcium indicator GCAMP in all fruitless positive neurons, including the sensory neurons in the male foreleg, and then developed a preparation in which we can basically image the axon terminals of these neurons as a male taps and tastes the pheromones on a female abdomen. And so what we found is that actually, if you look at the population of sensory neurons in the male foreleg, these fruitless positive population, in melanogaster males, these are strongly responsive to the taste of a melanogaster female and only weakly responsive to the taste of a simulans female, in accord with the idea that these neurons detect pheromones of an appropriate mate to promote courtship behavior. But remarkably, the tuning was almost indistinguishable in Drosophila simulans. And so what this indicates is actually in this species, in Drosophila simulans, they've dedicated this gustatory sensory population not to the detection of their own conspecific pheromone, but actually to the detection of the pheromone of an inappropriate mate to prevent courtship behavior. And importantly, in both species, actually, if we look in animals that are mutant for the PPK23 receptor, you see that these pheromone responses are lost. So this suggests that actually males of both species actually rely on the homologous sensory population that expresses the PPK23 channel to detect this pheromone. But these conserved sensory responses are really transformed into divergent courtship decisions. So we know actually quite a bit, as David beautifully described, about sort of the neural locus, a node within the courtship circuitry that is really integral to a male's decision to court. These were already introduced as the P1 neurons, which, as David mentioned, actually integrate from multiple sensory processing pathways and actually are able to dictate a male's courtship behavior. And so one way we can demonstrate this, which is very similar to what David just described, is that you could actually express channel rhodopsin in the P1 population, taking advantage of the really exquisite genetic tools that are developed for Drosophila melanogaster, and then provide the male with a slowly rotating magnet. And what you can see is the fly is basically completely indifferent to the magnet prior to stimulation. But then after even a very brief optogenetic illumination, the magnet becomes intensely attractive to the male fly, and he'll persistently pursue and follow it and sing to it. Despite that, obviously, that there's no appropriate behavioral feedback, the fact that the magnet does not reciprocate at all in his sort of ardent advances. So as David described, P1 neurons, activation of this population is sufficient to trigger a kind of persistent state of kind of social arousal. And we were actually interested to ask, given the fact that it will drive males to track kind of small rotating fly-sized magnets, what is the limit of their visual discrimination in this aroused state? And I will admit that these experiments were carried out mostly just from fun by Tom Hindmarsh-Sten, a graduate student in the lab. So what he found is actually these male flies, when P1 neurons are activated, are willing to chase house flies. And a ladybug, where the male fly actually gets on top of the ladybug before pursuing it around the arena. And even a lacewing, which is obviously morphologically much more different than, pardon me, morphologically much more, I apologize. It seems like my PowerPoint has crashed. I think I may need to restart.
It's not working.
Nope. I apologize.
You can continue.
Okay.
Well,
Okay, so I will,
we'll continue while it restarts.
so what we found actually, which I will just describe, is actually the fact that the P1 neuron activation of this population is able to elicit this kind of persistent courtship behaviors towards objects or morphologically very different insect species. And we're really interested to think more about what the limits of the visual discrimination are within this sort of aroused state, not using these kind of coarse methods of different insect species, but actually starting to use more refined methods. And one thing that we realize is that while these experiments seem really quite absurd, they also underscore a very interesting feature of courtship behavior, which is the fact that actually in the context of this aroused state, there's very little visual discrimination, but it really highlights the importance of these kind of inhibitory pheromonal cues to hone a male's desire towards females or appropriate mates. And this might be something that's actually particularly important in the context of natural courtship behavior, where male flies actually encounter a wide variety of different species in the context of their presentation. So let me get back to my slides, and then you can show you. Okay. Okay, so in the context of natural courtship, you can see that male flies actually show this arousal, general visual arousal, where they'll approach females of very different morphological body types, and then actually begin to sample their gustatory cues to assure that they are appropriate mate or not. So P1 neurons, we think then actually are sufficient to trigger courtship behavior in melanogaster males. So we could ask, what is the role of these neural population in simulins males? And to do this, we simply translated the genetic driver that labels this population in melanogaster into the simulins genome, allowing us to actually label a morphologically or anatomically very similar population of neurons. But most importantly, this genetic driver allowed us to then demonstrate that these neurons actually are sufficient to drive courtship behavior in a similar manner. So P1 neurons play a conserved role in promoting courtship across these two species. This led us to ask, what is the pheromonal tuning of this population? And so to do this, we developed a little preparation where we have a fly walking on a sort of air-supported ball, like a little fly-sized treadmill, and then we could bring in a female abdomen on a tether and allow him to sample her olfactory and gustatory bouquet. And so here you can see a movie of a melanogaster male that is about to taste the pheromones on a melanogaster female while we image the fruitless positive neurons, the neuropil where the P1 neurons innervate. And you can see as soon as the male taps and tastes the pheromones on the female abdomen, there's strong activation in this neuropil and specifically in the P1 population. But if we perform the analogous experiment in Drosophila simulans, we see that when he tastes the pheromones on a melanogaster female, there's basically no response at the level of the P1 population. And so what this suggests is that the opposing behavioral responses to sub-11-HD that exist across these two species are really mirrored in these kind of divergent responses at the level of the P1 population. And this seems to then provide a direct kind of neural correlate for the functional or the behavioral diversification across these species. But this diversification or the divergent P1 neuron responses are really, really in contrast to the conservation at the level of the sensory periphery that I described earlier. And so what this suggests is that there must be differences in the ascending pathways that link these kind of conserved functional responses to the divergent decisions that male flies make. So Josie Clowney, a previous postdoc in the lab, had really taken advantage of the exquisite genetic tools in melanogaster to really map out this circuit and to define its basic logic. And what she found is that PPK23 positive sensory neurons actually transmit their signals directly to an ascending pathway, which provides excitatory drive to the P1 population. But the VAB3 neurons also have a second target within the brain, a population of inhibitory interneurons termed MAL neurons, which are also poised to provide input to the P1 population. And so what you can see is that in melanogaster, this pheromone signal is processed through this bifurcating circuit architecture where there's direct excitation and disynaptic inhibition. And this kind of feed-forward inhibitory circuit motif is really kind of a canonical motif that is found ubiquitous throughout many nervous systems, where it is really thought to be integral to gain control, because inhibition will be directly proportional to excitation. And we think it plays a very similar role in the context of the courtship circuits in melanogaster, where it provides stringent regulation of the P1 neuron excitation to assure that males don't inappropriately and inadvertently enter this kind of hyper-aroused state where they're willing to court completely inappropriate inanimate objects. So having mapped out this circuitry, we can now ask really what is the, compare the ascending pathways in Drosophila melanogaster and simulins. And what I'm showing here at the level of function, but not at the level of anatomy, is that actually in both cases at the level of anatomy and function, that there's really conservation throughout this pathway all the way through to the level of the P1 neurons. And so while there might be multiple, or could have been multiple changes that occur at each level of the sensory processing pathway, we think that the P1 neurons serve as a kind of a key node for functional differentiation. And in particular, the integration of these two opposing pathways, the excitatory branch and inhibitory branch, by simply reweighting the inputs to these two on the level of the P1 neurons, can allow for different responses across these two species. Such that in melanogaster, when a male fly tastes the melanogaster female pheromone, it drives excitation of the P1 population to promote courtship behavior. But in simulants, there's been a reweighting of the inputs such that inhibition dominates. And so when he tastes the pheromones in a melanogaster female, that actually drives inhibition of the P1 population and suppresses courtship behavior. And so together, this data suggests that actually the species-specific kind of behavioral responses to sublumin HD that serve to erect a pre-mating reproductive barrier between these species, we think are built not from changes at the level of the sensory periphery, surprisingly, but really due to a central circuit switch in which actually the kind of reweighting of inputs at the level of the P1 population transforms an excitatory pheromone that promotes courtship behavior into an inhibitory pheromone that suppresses courtship behavior. So one of the really interesting implications of this circuit is the fact that actually, as I mentioned, it is in simulants, this pathway is entirely dedicated now, we think, to actually the detection of pheromones of an inappropriate mate. And while I showed you that P1 neurons promote courtship behavior in both species, we find no evidence that there's excitation of the P1 neurons, even to the taste of simulants pheromones. And in some ways, this makes some logical sense that actually pheromones serve a primarily inhibitory role. In part due to the fact that actually pheromones in simulants are sexually monomorphic, that is, males and females make the very same compounds that cover their cuticle. And so these would serve as ambiguous cues for mate recognition. But this also requires then that there must be additional sensory pathways that are integrated by the P1 population, ultimately to promote the male's arousal. And we think that vision might be playing one such role. So together, this begins to underscore, though, why integrative nodes like the P1 population might actually serve as a very flexible substrate for behavioral evolution. Because really, if you think about it, all males actually need to identify an appropriate mate and make the decision to court, but they might all do so with a distinct complement of excitatory and inhibitory cues that are species-specific. And in that sense, we're interested to think about the idea that P1 neurons might serve as a kind of a node for flexible behavioral variation across species, this kind of integrative nodes, in a similar manner to the idea that actually there are certain genetic nodes where actually repeated evolutionary tinkering has given rise to the diverse morphological patterning that exists in the animal world. For example, repeated evolutionary changes in the cis-regulatory element of the yellow locus has given rise to this kind of beautiful variegated pattern of pigmentation in the wings of different dipterians. And so to gain kind of broader principles of behavioral evolution, we're now extending our analysis to broader branches of the phylogenetic tree in Drosophila, taking advantage of the fact that actually the pheromones, female pheromones, have rapidly diversified across these different species due to the rapid evolution and repeated and independent evolution of the biosynthetic enzymes that produce these different pheromones. And so if we even just look at the three close cousins to Drosophila melanogaster, Drosophila simulans, Yucuba, and erecta, all of these three females in these three different species actually make a compound that has been independently evolved and is distinct from the melanogaster pheromone. And accordingly, all the males of these three different species have derived an aversion to the melanogaster pheromone sublumin HD and actively avoid courting their own conspecific females, which are actually perfumed and tainted with the melanogaster pheromone. And so this offers us the opportunity then to ask, are there common or distinct mechanisms for this kind of parallel behavioral evolution? And so this is something we've just begun to address by extending the neurogenetic toolkit into these more distant species. And this is the work of Rory Coleman in the lab and Nessar Morante with the help of David Stern from Genelia. By labeling the fruitless positive neurons in Drosophila, Yucuba, and erecta to gain access to the same pheromone processing pathways, but also extending our cell type specific driver lines to these more distant species. And for example, this now allows us to ask what is the role of the P1 neurons even in these more distant Drosophilids. And what we can do then is to demonstrate by optogenetic activation using the specific driver lines so that actually activation of the P1 population in these distant species is sufficient to drive courtship towards inappropriate objects, be them magnets or ladybugs. So P1 neurons play a conserved role. They serve as a conserved kind of node in the courtship circuitry across many different Drosophilids, allowing us to start to think about their pheromonal tuning and how pheromones shape their responses. And so these are ongoing experiments, but I just wanted to highlight a few interesting principles that we started to reveal by looking at this kind of broader phylogenetic comparison. So if we look actually at species that are sexually dimorphic, for example, Drosophila melanogaster and its more distant cousin Drosophila erecta, what we find when we image the neuropil where the P1 neurons innervate is that these the P1 neurons appear to be selectively tuned to the pheromones of their appropriate conspecific female. So these neurons are strongly activated by their own appropriate female pheromone, suggesting pheromones really provide an instructive signal to promote courtship behavior. But interestingly, if we look at species like Drosophila jacoba, they are just like Drosophila simulans in that they're sexually monomorphic in their pheromone production. Males and females carry the same pheromones. And we see in this case that actually pheromones serve a completely, we think provide a completely inhibitory role, and are, even the conspecific female pheromones are unable to activate this P1 population. And so what this suggests is that there are really multiple distinct strategies that different Drosophila species are using for mate discrimination based on pheromones. And we're interested now to start to tease apart the kind of different neural adaptations that occur to see whether these sort of parallel changes at the level of kind of pheromone usage and mate discrimination are implemented through the same kind of central circuit switch that I described for Drosophila simulans, or actually does every species actually has evolved its own unique kind of solution to the same sort of computational problem. And so I just want to end by actually really highlighting, I think from my perspective, that this is a very exciting time for comparative neuroscience because of their ability to really begin to gain access to kind of homologous circuits between closely related species and actually circuits throughout the kind of animal kingdom. And I think just as kind of studies of amino acid sequence homology have been absolutely instrumental into our understanding of protein mechanism, by taking these kind of comparative approaches, we hope to be able to tease apart ultimately which are the central and kind of core circuit motifs that are fundamental to all nervous systems from the kind of idiosyncratic adaptations that are tailored to any particular species. And so with that, I would just like to acknowledge the people in the lab who carried out this work and actually advice and input from David Stern and others at Janelia. And I'd like to thank you again for the invitation to be here.
Fantastic, Vanessa. Again, questions? I note one there, but please, for the young fellows, I will come after you sooner or later if you don't pose questions. So, one there and one here with David. Please.
Thank you for just a beautiful science, a beautiful lecture. I just wonder, from evolutionary perspective, does it really make sense to develop a system that makes you basically court your siblings and cousins rather than diverse your genome? What was the evolutionary benefit of that?
So you're referring to the fact that they're not cousins, meaning related within the individuals within a species, but very closely related species. So,
and that was a later development. So rather than diverse the genome, you make it more specific.
So it's always interesting to think about why these systems may have evolved. Yes. And I don't think we have actually obviously answers to it, but I can only speculate. And I think one of the things that's interesting is it is sort of an inverted logic that you might, and I think what most of us had anticipated, where you might imagine females make an instructive cue that's species specific and all the males are attracted to that one cue. Instead, it's actually the inverted logic where basically males are generally aroused and they use these pheromones to prevent courtship towards inappropriate mates. And in some sense, it could be, there are a couple reasons that may be the way the system emerged. One reason is that actually it could be evolutionary more flexible, in part due to the fact that if either the receptors or the pheromones change, that actually males, instead of being unable to be attracted to a female, are now just basically promiscuous. The other possibility is actually the chemistry of these pheromones in Drosophilids are actually quite constrained, partially because of the fact that they actually coat the female's abdomen or the cuticle, and it serves as an alternative adaptive element to prevent desiccation. And so it could be that is one other thing which limits the kind of diverse repertoire of chemicals that are being used.
David?
In melanogaster, 7-trichosine plays a role on males to prevent male-to-male courtship. And it's also on males in simulans, but it can't play that role because it's on female simulans as well. And otherwise, it would prevent male-to-female courtship. So it seems as if responses to 7T have co-evolved between simulans and melanogaster in parallel with responses to 711HD. And I wonder if you have any insights or data onto the way in which that evolution has occurred.
That's right. So we've looked only at the role of the kind of evolution of the responses to 711HD, but it also requires that there are differences between melanogaster and simulans even for their perception of 7-trichosine, which in melanogaster males is inhibitory and in simulans males must be neutral. We actually don't yet know fully what the mechanistic basis for that is. But it's also interesting to think about actually then how is sex discrimination achieved within these species that are sexually monomorphic. And so one interesting idea is that additional cues, like for example, the volatile pheromone cis-fascinoacetate might play a much more important role. Again, sort of highlighting how the different complement of cues might be reweighted at the P1 population in these different species.
Question there?
Thank you so much for the fascinating talk. You showed a very strong, attractive response from a male to a female of the same species, as well as a very strong aversive response from a male to a female of a different species. I was wondering, within females of the same species, is there a fine-tuning of cues of whether this female is an attractive or aversive mate? And do you think that this relies on the same flexible P1 neurons?
So are you asking about the circuitry in the females, essentially, or?
I'm asking about, for a female of the same species, is there always an attractive response from the males, or can a female of the same species be aversive, and what circuit might that rely on?
Yeah, so there is additional layers of kind of regulation, probably at the level of the male. In one way, for example, actually David has beautifully shown that past sexual experience, social experience, affects the excitability of the P1 neurons, which is actually another interesting idea that actually both kind of evolutionary adaptations and experience-dependent adaptations might occur at the same sort of central circuit site. So that's one way actually males actually probably regulate their courtship behavior additionally.
Thank you so much.